Well, great. It's an honor to be here. Thank you so much for taking the time to hear what we're doing here at Senti. So Senti is a company based up in South San Francisco focused on creating a platform for programming cell and gene therapies with increasing precision, control, and efficacy. Senti is now a public company, so we'll be making forward-looking statements today. So what we're doing at Senti is essentially building a platform for applying synthetic biology to the cell and gene therapy space. We believe that with tremendous advancements in new modalities and manufacturing, we now have so many different types of cell and gene therapies that we can push into the clinic. However, we believe that if we want to take these technologies and really make a significant impact in wider patient populations where the targets may not be as clear or we need to improve the controllability or activity of these products, a significant improvement in how we program these cells is going to be quite important. And so core to what we're doing at Senti is really being able to genetically program and design regulation and targeting into these products. So these are done through what we call gene circuits. These are multi-gene constructs that we've designed that can interact with each other in a specific way to give rise to different functionalities. They include multi-arming, so building cells that have multiple functionalities. They include logic gating, so building cells that can sense and respond to multiple targets, not just a single one. They include regulation, so we can dial up and dial down the activity of these products with FDA-approved small molecule drugs. And they include sensors, basically sensors that we can insert into these products so that they can sense their environment and tune their activity as a response. Internally at Senti, we've built a pipeline of CAR-NK programs that are allogeneic based off of healthy adult donors. We have process development and manufacturing that allows us to scale and produce these in-house. And we're targeting both blood cancers such as AML as well as solid tumors with these approaches. We've also established 2 platform collaborations, one with Spark around AAV design, where we're creating customized promoters that give very selective activity in certain on-target cells while minimizing off-target expression, as well as with BlueRock building in regulatory circuits into iPSCs for regenerative medicine applications. As a team, we're very happy since the last time we were here to bring on Dr. Kanya Rajangam as our new Chief Medical and Development Officer. She's built up a wealth of experience working on allo cell therapies, especially in AML, which is one of our lead programs. So I thought I would start today by giving you guys an overview into what we're doing on the CAR-NK side to give you a sense of what these gene circuits can potentially do for us. There's been a lot of advances in the development of cell therapies as well as NK cell therapies specifically for oncology applications. We think there are several major challenges, though, that still face us in the field. Number one, how do we further improve the activity, the persistence, and the expansion of NK cells? And we've been exploring the use and engineering of cytokines as well as other payloads that give us multifunctionality in the NK setting. 2 is a general challenge with all targeted therapies. How do we deal with antigen escape or tumor heterogeneity? Can we target more than one target at a time to try to increase the spectrum of killing? Number 3, we think, is a central problem with any ADC, bispecific, or single target CAR. If you don't have a clean target, you're always going to be trying to thread the needle between affecting the healthy cells versus trying to kill the tumor. So with our logic gates, specifically a type of logic gate called the NOT gate, we can now separate or solve that problem by protecting the healthy cells explicitly with a protective antigen while still maintaining good on-target on-tumor killing. And that's through the design of an inhibitory CAR that allows us to essentially mitigate NK toxicity or any CAR toxicity against healthy cells. Finally, a lot of these efforts we're taking to engineer the cells, to express cytokines, to regulate their activity, et cetera, to change that tumor microenvironment. We want to be able to control that process so that we can turn it on or off on demand if we need to try to better regulate what these cells are doing in vivo. We can do that through what we call the regulator dial, which we've used to control cytokines like IL-12. I won't mention too much sort of the reasoning for CAR-NK since I think we heard this in a previous talk, but we believe by being able to go after indications that have dirty targets with our logic gate technology, by being able to engineer multifactorial payloads into our NK cells, we have the ability to start tackling some of the central challenges in the CAR immune cell space. So let me highlight one platform technology that's going into all of our programs, which is basically engineering payloads that are produced by our cell therapies that are not only on the cell surface, but also secreted. The way we do this is through a technology called calibrated release IL-15. In the upper left-hand corner, essentially what we do is we can attach a cytokine or some other immunostimulatory payload to the cell surface, and then that is cleaved off by specific proteases. And so that allows us to release a soluble fraction of the active molecule that allows us to achieve paracrine stimulation, which we can see in the lower left-hand corner. We can get essentially equivalent stimulation of STAT signaling in PBMCs using this approach compared to a soluble IL-15. We also get the membrane-bound version of IL-15, which we've shown also to be very important to enhancing persistence, which you can see in the upper right-hand corner, as well as enabling repeatedly good serial killing assay. So on the lower right-hand side, this is an assay where we've essentially re-challenged the cells multiple times over a 200-plus hour time period. And we can see that in blue with the calibrated release IL-15 construct, we were able to achieve significant killing across all of that population. Compare that to, for example, in pink, using a secreted IL-15 alone, we see after the first round of killing, essentially these cells lose their potency. So we've been able to apply this platform not just to IL-15, but to other cytokines. Any cytokine where you want to leverage both the paracrine and autocrine nature of its ability to signal. And we can even do this in combination. So how are we applying our platform? We're building a pipeline of programs, starting with Senti 202, which is an AML-focused program, going after CD33 as well as FLT3 as the tumor targets. And we've designed in a NOT gate in that case to protect the healthy cells from being killed. That same logic gating approach is being applied to Senti 401 for colorectal cancer and other CEA-positive tumors, as well as other solid tumor programs we haven't disclosed yet. Senti 301 takes a slightly different tack and really incorporates our ability to arm cells with cytokines to enhance their activity for solid tumor indications. So what I wanted to do is give you guys a bit of a flavor of what we can do with these logic gates, and starting with Senti 202. Senti 202 contains essentially three major components. The one on the top is an activating CAR that simultaneously targets FLT3 as well as CD33. So it's designed in a bivalent format. We chose the two particular targets. They're pretty well known in the AML field, but they do cover different subpopulations of the AML tumor burden. CD33 tends to be found more on the blast population, whereas FLT3 can be found on many of the leukemic stem cells. So by being able to target both of those, we think we can significantly elevate the anti-tumor killing activity of the product. However, CD33 and FLT3 are also known to be expressed on healthy cells, including myeloid cells and healthy hematopoietic stem cells. And so in order to mitigate the risk for bone marrow suppression and to try to widen the therapeutic window, we've designed the element in the middle here in purple, which is an inhibitory CAR. Basically what it does is it recognizes a protein that is highly expressed on the healthy cells, in this case the healthy hematopoietic stem cell, but is not expressed on the cancer cells. And so it essentially serves as an artificial do not eat me signal. In this particular case, this NOT gate CAR receptor recognizes a target called endomucin. And we discovered that through bioinformatics and validation on specific patient samples. Finally, the bottom right component includes the calibrated release IL-15 I mentioned earlier. So we have shown in a variety of data, in previous conferences, I only sort of pull out a highlight here, but we've shown that essentially Senti 202 does retain good functional killing activity across a pretty wide range of AML cancer subtypes, as well as in a variety of AML models shows anti-tumor activity. The logic gating also functions both in vitro as well as in vivo. Essentially, the way we characterize this is we've taken primary human HSCs, and we basically try to characterize, does the NOT gate essentially protect these from killing? So on the left-hand side, this is an in vitro assay, relatively short time point. But basically, on the left-hand side of this graph, we show that the Senti 202 CAR NK cells that contain the NOT gate are able to functionally kill AML cancer cells, similar to NK cells that don't have the NOT gate. However, when you expose them in the middle to hematopoietic stem cells, essentially you see a reduction in killing. This ability to discriminate is significantly amplified for longer time periods as well as in vivo. So on the right-hand side, this is an in vivo model where we essentially inject both healthy cells and cancer cells into that model, followed by treatment with the CAR NK. And you can see that on the right-hand side in blue, we see a significant enrichment in the blood of those healthy cells, and and the concomitant loss of the cancer cells in this situation. We are quite excited about the potential for this in CD33 positive and FLT3 positive malignancies. We are going to focus on AML as a major indication for this, but we do think there are opportunities to expand this into, for example, MDS, as well as other heme malignancies that express these particular targets. This concept also extends into the solid tumor setting. So I think one of the big challenges with tumor-associated antigens for solid tumors is many of them are also expressed on healthy tissues. And again, that's going to limit your ability to treat these patients aggressively, potentially. So in this particular situation, we have developed a CEA-based product on the upper left-hand side, CEA targeting activating CAR. CEA is also found on healthy epithelial cells, including in the lung and on the healthy colon. So in this particular case, we repeated our discovery approach, discovered a protective antigen called VSIG2, which is found on those healthy epithelial cells, but not found on colorectal cancer samples. And so that's been used here to also develop this inhibitory CAR element that can protect those cells from being killed. In this particular case, we also have the calibrated release IL-15, as well as a fourth undisclosed element that further synergizes with the IL-15 activity. Just to show you one additional piece of data here, and we will have more coming out at SITC soon. But basically, if you look at VSIG2 staining, you can see that there's a pretty big differential in terms of VSIG2 expression between healthy colon epithelium and colorectal cancer cells, whereas CEACAM5 is expressed in both those tissues. So this, I think, hopefully clearly illustrates the need for a NOT gate to try to protect these healthy cells and maximize your therapeutic window. On the upper right-hand corner, we have shown that CEA CAR NK cells outfitted with just one of those payloads, the calibrated release IL-15, is able to achieve, again, repeat serial killing. We have now also translated this into in vivo models. And on the bottom right-hand corner, we've been able to show that we can take the NOT gate and essentially readapt it to a different target. In this case, we can show the significant protection of CEA-expressing cells that also express VSIG2 in this case. Finally, the Senti 301 product goes after GPC3, which is a target in liver cancers and some other solid tumors. It contains our calibrated release IL-15 construct, as well as the ability to regulate. We have the ability to add a component here that allows us to regulate the expression of other cytokines, such as IL-12. We have shown that the calibrated release IL-15 GPC3 construct does have a potent preclinical anti-cancer activity, both in effective in vitro serial killing, as well as increased survival in a variety of animal models. And this is without the IL-12 component. The IL-12 component is actually quite interesting. IL-12 is just one example of a very potent cytokine that we would like to express to transform the tumor microenvironment. IL-12 has been tested in the past as a recombinant protein. Clinical use has been limited in part due to toxicities, a narrow therapeutic window. And so what we've been able to do is actually regulate IL-12 secretion using a variety of different small molecule drugs. In this particular case, I'm showing you an example using grazopavir, which is an FDA-approved hepatitis C virus protease inhibitor drug. And what you can see from this data is that we can regulate in a dose-dependent fashion IL-12 expression over 100-fold on-off, and that this is a reversible system. We can turn it on at day 2, have it go away by day 9, and turn it back on again. So this ability to titrate and regulate, we think will be quite powerful in the future in going after solid tumors and really try to maximize the ability of these cells to try to change the tumor microenvironment with really potent payloads. In the remaining time, just want to give you a highlight on other places we can use our technology. We have a core synthetic biology gene circuit team within the company that essentially is the brainchild for all of these different programs, whether it's external or internal. And the reason we've designed it that way is that really allows us to build synergy and efficiency in designing these constructs for any given application. Our collaboration with Spark is focused on AAV, designing promoters that are specific to certain cell types, and our collaboration with Blue Rock is focused on the regenerative medicine space. Just to give you guys a taste of sort of some of the data that we've generated, for the AAV application, we are very focused on building highly compact synthetic promoters that are targeted to your on-target cells, have minimal expression in your off-target cells, and are able to be tuned in terms of their strength. Right? We don't just want specificity, but we also want to be able to control the strength related to this. And so we think this is going to be important as gene therapy tries to expand into broader indications where you may not have enough specific tropism with the vector alone. Can we mitigate the off-target tissue tox, and can we also improve potency of the product? This bottom right-hand corner is showing you a library of promoters that we've been able to create. On the y-axis is specificity, which is calculated by the ratio of expression between your on-target and your off-target cells. And then we have strength on a relative scale on the bottom. The one arrow there points to a constitutive CAG promoter, which is a known promoter in the field. And what you can see here is we have promoters in the upper right-hand corner here that are over 10,000-fold specific, but actually even stronger than CAG. So we can actually design promoters that have sort of both specificity and strength built into them as desired. So in summary, Senti is a platform company really focused on designing gene circuits for all cell and gene therapies of the future. Internally, we're focused on NK. We believe there's a tremendous product opportunity there in oncology to demonstrate the utility of our genetic technologies. But we also think these gene circuits are very portable to T cells, to gene delivery vehicles, other cell types. And so Senti is very open to partnering with companies that have interest in taking those types of programs forward in collaboration with us. So with that, thank you very much, and hope you have a good rest of the conference. Appreciate it.